Okay, DNA replication has mitosis and that's
Look at D in the picture. Do you see where it says the two centers are in Victoria's? So let's keep the next G2 after this. It's hard to sell.
Okay, so let's talk about this a little bit. One of these is probably really obvious. Which part of the cell cycle do you think is the shortest amount of time? Yeah, the M part, which it's drawn that way in this diagram, and it kind of makes sense. The actual process of dividing the cell doesn't take a very large portion of the cell's life cycle. The other three are a little bit trickier, partly because this diagram is not totally drawn to scale in terms of time. So if you're judging by the length of the arrows, that's not the best way to make the judgments on this, but I didn't tell you that, so that's okay. Think about what happens during each of these phases. Out of the G1 and S and G2 phases, which one do you think is the most important and requires the most precise work within a cell? Yeah, DNA synthesis is this S phase. I heard at least one or two groups say that. That's actually the longest part. So the 10 to 12 hours would be spent on DNA synthesis. That's the part where the DNA is actually being replicated and copied within the cell. So that takes a while. That genetic information is really important and needs to be copied accurately. And there's a lot of it, and all of it needs to be copied. So that process of DNA replication takes the longest part of a cell's life cycle. The other two sections, the G1 and G2, are pretty similar in length and can also vary a lot. G1 is generally a little bit longer, but not always, and that varies a little bit more because those parts are just kind of in between when cells are growing and kind of checking what's going on. So that's just to kind of give you an idea of roughly how these go together. So we're going to go and talk a little more detail about what's happening in each of those parts. First of all, it's important to realize, and you can kind of see from the colors on this slide, G1, S, and G2 are all part of interphase, which is just kind of like the everyday life of a cell when it's not actually in the process of dividing. Interphase makes up most of the life cycle of a cell, and it's divided into the G1, S, and G2, but that's all interphase. The M part stands for mitosis, or the mitotic part of the life cycle when the cell is actually dividing, okay? So during G1, the G actually stands for gap because when scientists first saw cells in these parts of the cell cycle, they didn't think they were really doing anything, so they called it kind of a gap in the cell's life. But I usually think of the G for growth because especially during G1, after cells divide, the two new little daughter cells would start right at the beginning of G1, and the first thing they need to do is they need to grow. And they need to produce more organelles, and they need to, of course, carry out all of those cellular functions that cells do. So the G1 is just a basic time of growth and metabolism. At some point during G1, a cell needs to kind of make a decision, and I use that word not that cells have a brain, but it's a decision based on the chemical signals that it receives, which we'll talk about more later this week, whether or not it's going to divide. Because a lot of your cells in your body don't need to be dividing all the time, right? You are not constantly growing. When you have a growth spurt, you grow a lot, so your cells divide more, but then you might stay the same height for a while. Or like me as an adult, I'm not growing all that much. My cells only really need to divide when they're healing something or repairing or replacing old cells. So the vast majority of your cells in your body aren't actively dividing. And so a lot of cells are just kind of in this holding pattern. If a cell does not need to divide, it goes into what's called the G0 phase, which is just kind of this little side loop on the cell cycle. Some cells just enter the G0 phase temporarily. And then if you get injured or if you need to grow again or something like that, they can move back into the rest of the cell cycle and continue to divide. Some cells move kind of permanently into the G0 phase, and once they are fully mature cells, they're not going to divide anymore. So that can be either a temporary or a permanent thing, but it's just kind of a holding pattern for cells that are not about to divide. Because once a cell enters the S part of interphase, S stands for synthesis, because that's the part where the DNA gets copied. That's where DNA replication occurs. And so as soon as a cell starts duplicating its DNA, the reason it does that is because that's the first step of preparation for cell division. So if the cell doesn't have to divide, it doesn't copy its DNA yet. It only does that if it's moving towards division. And then the G2, that second gap, is another period of growth and metabolism, but that's also when the cell kind of goes through and double checks and proofreads the DNA to make sure that it's copied correctly and fixes as many of the mistakes as it can so that they don't become permanent mutations that get passed on to cells. So all of that is part of interphase. And during this whole time, G1, S, and G2, if you look in a microscope, you can't actually see the chromosomes visibly because the DNA is just kind of loosely spread within the nucleus. It's wound around some proteins, but it's not real tightly coiled up, so it's not really visible. The only time the chromosomes coil up tightly enough to become visible are during the actual process of cell division. That's a way that cells kind of organize the chromosomes, organize that genetic material so that it can be separated and divided neatly. So that last part is mitosis and cytokinesis. So the actual division of the nucleus and division of the rest of the cell. Questions about the basic cycle? Okay, so let's go through and talk about kind of what's going on in the cell in each of these phases. And some of the different parts. So you'll see the pictures that you sorted yesterday as we go through here. I'm going to add some labels. If you labeled things somewhere else, that's okay. But make sure you're labeling the right thing. So you can kind of double check yesterday's work and you can add some of these notes in your packet, whatever works for you. So interphase, like I said, is made up of G1, S, and G2. During this time, the centrosome also duplicates. That happens during the S phase as well. The centrosome is this region down here. Oh no, pause. There we go. So this region down here, better, is called the centrosome. It's a region of the cytoplasm. So it's an area within the cytoplasm that contains centrioles if this is an animal cell. And it also organizes some of the cytoskeleton. It organizes structures called microtubules that are important in this process. So, remember, centrioles are an organelle that only exist in animal cells. Plant cells do not have centrioles. But plant cells do still have a centrosome region within the cytoplasm that helps to organize that structure, that part of the cytoskeleton. The other thing that happens during interphase is that the chromosomes duplicate. Like we said, DNA replication happens during the S phase, but they are not visible. The DNA at this phase during interphase exists in a form called chromatin. And chromatin consists of DNA and proteins. The DNA is kind of loosely wound around those histone proteins. If you remember, we looked at a diagram in the last unit, I think, that showed this kind of like beads on a string. That's kind of what this looks like. So it's wound around the histones, but it's not coiled up super tightly, and it's not visible when you look through a microscope. At the beginning of prophase, one of the first things that happens is that the chromosomes condense. That loosely spooled chromatin starts to wind up tighter and tighter into these structures we call chromosomes. And so we're going to talk a little bit about those chromosomes. Remember, by the time these become visible, they have already duplicated. They already went through DNA replication. So this picture, and you guys have this on a slide, right? Yes. This picture shows one duplicated chromosome, okay? So that's what this is, and we're going to label a bunch of stuff on it. And the words might show up right underneath that. We'll see. So that chromosome, since it has already duplicated, contains 2 copies of the same information. And each of those copies is referred to as a sister chromatid. So that's this first thing we're labeling. So you can see on my slide, one sister chromatid is light purple and the other one is dark purple. Those are 2 identical copies of the same DNA code. There are proteins that attach those 2 sister chromatids together called cohesins. If you know what the word cohesive means, it means things kind of work together, they stick together, right? So cohesins attach these sister chromatids to each other right through the middle. And actually, it's only shown connected in the middle, but those cohesin proteins would actually hold it together along the length of the sister chromatid. So those are those cohesin proteins, kind of those little blue circles I just drew in there, that attach those 2 sister chromatids all along the length of it. The place where they are most tightly connected, though, is called the centromere. And the centromere is actually the region of the DNA. So it's this part, here, I'll grab red. It's this part kind of right in the middle, this region of DNA through here is the centromere. Like I said, those cohesins do attach the sister chromatids along the length, but they are the most tightly and completely attached to each other in that centromere region, which sometimes is right in the middle, like in this picture. Sometimes the centromere is located a little further up or down on the chromosome. It's not always immediately in the middle, but it's often drawn that way. Then you'll also notice right around that centromere region, there are these 2 kind of brownish round things sticking out. Those are called kinetochores. And what those are is a complex of proteins that is where the spindle, those parts of the cytoskeleton, it's where those microtubules of the cytoskeleton attach to these chromosomes. And later, that's what will actually pull these 2 sister chromatids apart from each other. there's a kinetochore on each of the sister chromatids. There's that complex of proteins on each one that attaches to the spindle microtubules. That's these little lines coming out here. That's actually part of the cytoskeleton that's attaching to those proteins that helps to move these chromosomes where they need to go during the process of mitosis. Any questions about any of these structures? Okay, can I erase all this text? Yeah? Okay, so then we move into prophase. And in your pictures, you kind of had a few images that represented prophase, because remember, this is a continuous process. The top one up there would maybe be very early prophase when the chromosomes are just starting to become visible. Usually we draw prophase more like the middle one, where you can see those duplicated chromosomes clearly, but they're not really organized anywhere. And then even when they kind of start moving more towards where they need to go, that's still prophase because we haven't quite gotten to metaphase yet, okay? So during prophase, the chromosomes are visible. This is also when the spindle begins to be built. Prophase is really the time of preparation. And so the spindle, think of the spindle kind of like scaffolding that's built during the process of mitosis. It's a temporary structure that helps move the chromosomes where they need to go, just like scaffolding is put up during a construction project, but then once the construction is done, the scaffolding is taken down and goes away. Same thing with the spindle. The spindle is kind of a rearrangement of the microtubules in the cytoskeleton just to help with this process of division. And then once division is done, the spindle breaks down and some of those proteins can be reused to build other parts of the cytoskeleton. Okay, so the aster, the word aster literally comes from a word that means star. So it's this early form of the mitotic spindle that looks like just a little starburst. That's the aster. Also during prophase, the centrosomes begin to migrate to the two ends of the cell. Sometimes we call those the poles of the cells, like the North and South Pole. But they start moving away from each other. Normally, those centrosome regions are found pretty close to the nucleus. But when it's time for division to start, they migrate to the ends of the cell to help organize that spindle. Because remember, those centrosome regions are what set up this arrangement of microtubules and organize them in that way. The other thing that happens here is that the nuclear envelope breaks down. During the process of mitosis, these chromosomes need to be able to move within the entire cell, and so that membrane that surrounds the nucleus breaks apart, and those lipids are then just in the cytoplasm so that they can be reused again later. Questions on this part? Then we move into metaphase, probably the most easily recognizable of the phases. As the chromosomes start moving throughout the cell, they attach at those kinetochores to the spindle. And so like you saw in that chromosome diagram we were just labeling, the microtubules of the spindle attach to each of the sides of that chromosome, to each sister chromatid. And as they do that, they kind of tug on the chromosomes. And that's actually why the chromosomes end up lined up in the middle, because when they're attached on both sides and they're being pulled equally in both directions, they end up just hanging out right in the middle of the cell. So that's why the chromosomes line up in the middle during metaphase. And that's what that one says. They line up in the cell along an imaginary line called the metaphase plate. That's what this dotted line represents. It is very important to realize the metaphase plate is not a real thing. It's kind of like the equator, right? The equator is a line that we drew on the map, but if you go to the equator and look at the ground, there's nothing there, right? The metaphase plate is the same thing. It's an imaginary line that we make up in our heads to help us kind of show where the chromosomes line up. It's not an actual structure within the cell, okay? Metaphase happens really quickly. The spindle, as soon as the spindle is attached on both sides to all of the chromosomes and they're lined up in the middle, we move on to the next part, which is anaphase. In anaphase, you can remember A for away because the chromatids start to move away from each other. Those cohesin proteins that held the sister chromatids together are cleaved or broken apart, sliced through by an enzyme called separase. It separates the two sister chromatids. And each sister chromatid, as soon as they're separated, is considered its own chromosome. When they're connected to each other, they're counted as one chromosome. As soon as they separate, they're considered two, okay? A special type of protein called a motor protein drags these chromosomes back by walking along the microtubules. And these are actually my favorite proteins, like in all of biology. I might have shown you this before. I think this is the one I want to show you right now. There's no sound on this animation. But this is like a computer-generated animation of a motor protein. This one's carrying a vesicle instead of carrying a chromatid. But they're actually proteins that look like they're walking. It's not alive, it's not walking. It's a series of chemical reactions and changes within the shape of that protein, which is kind of amazing. Yeah. Hang on, let me escape out of that now. If you're curious about how a protein can do that without actually being a little alien walking along in your cells, if you go into the slides and click on this word, there's like a 2-minute video that actually talks you through a little more of the chemical changes happening in that protein that change its shape such that it attaches and then releases and attaches and releases to allow it to make that walking movement. But if you ask me what my favorite proteins are, that's probably them because I think they're cool. I know, I do like little aliens walking through your cells too. Okay, also during anaphase, the other thing that happens, so some of the microtubules that make up the spindle are what are called kinetochore microtubules. Those are the ones that are attached to the chromosomes. And those are the ones that are basically acting as highways for those little motor proteins to walk the chromosomes back to the two ends of the cell. But the other microtubules that are not connected to the kinetochore are called non-kinetochore microtubules. During this phase, they get longer. They actually get built longer and more proteins are added to them. So that would be like this one right in here, and this one through here, and this one that don't have chromosomes attached to them. They start to lengthen because what that does is it elongates the shape of the cell and basically makes enough room so that as we get to the next stage, we can start dividing the cytoplasm and the cell membrane and all the rest of that. Once the chromosomes have separated and moved out to the ends of the cells, we get to telophase and cytokinesis. And these two phases overlap each other. Telophase is when a nuclear membrane reforms around each group of chromosomes. So you have a cell with 2 nuclei in it. And it's the end of the division of the DNA and the nucleus. And mitosis is technically division of the nucleus. Cytokinesis is the division of everything else. And cytokinesis happens a lot faster because it doesn't have to be nearly as precise. The division of the DNA is very structured and very, first we do this, then this, then this, because it has to be done perfectly. But it doesn't matter to the cells if they have exactly the same number of ribosomes in both the daughter cells and exactly the same number of lysosomes and whatever else. That doesn't matter because the cells can make more. So this part happens a lot more quickly. Cytokinesis is slightly different in plants and animals because plant cells have a cell wall and animal cells don't. The way that they divide is slightly different. In animal cells, that's the picture on the left here, a ring of microfilaments, that's the same structure or another structure that makes up the cytoskeleton. A ring of those microfilaments forms around the center of the cell, and then it just slowly gets shortened and starts literally squeezing the cell in two because the cell membrane is flexible, and so it can kind of just be pulled in like that. And if you look at a cell under the microscope in anaphase or telophase, sometimes you can see the beginning of that shortening, that little separating of the two cells. That's called the cleavage furrow because that's where it's going to cut those two cells in half. So animal cells are pretty simple. They just kind of get squeezed into two, kind of like if you have one bubble and it just separates into two. Plant cells are a little bit harder because they have to build a cell wall in the middle as well. And so plant cells actually have vesicles that form that carry proteins and other molecules that build the cell wall. Those proteins and other molecules are made by the ribosomes and the rough and smooth ER. They get packaged by the Golgi, that's the organelles that you see in this picture on the right. And those vesicles bud off from the Golgi and travel to the center where they start releasing those molecules to build the new cell wall. The early version of that structure is called a cell plate. That's basically a new cell wall in progress being built. So that eventually divides this into two new cells and finishes off cytokinesis in those plant cells. Questions? All right, good deal. So the rest of today is going to be some more work time for you. If you need to go back, now that we've kind of talked through it, if there's anything you didn't know where to label it or you put it in the wrong spot yesterday, feel free to go and fix that. Then the other activity in the cell cycle activities post is called the cell cycle regulation packet. Please start working on that. If you don't 100% get through it today, that's okay, but I'd like you to have it pretty much done by Thursday, which is when we'll really start talking about the topic of how this cycle is controlled and how cells know when to go on to the next phase and when to just kind of hang out for a while. We are going to have an assessment tomorrow. The assessment tomorrow will mostly focus on biotechnology and the stuff we did last week, with just a little bit of basic stuff about the cell cycle and these different phases we just talked through. Anybody have any questions? So I think what we're going to do for tomorrow, just because it's easiest for me and my brain literally can't think of anything new right now, I think it's going to be a College Board assessment. And if you're sitting in the room, you will just go on during class and take it then. Like I will just post it and open it at class time. And people at home will do the same thing. They'll report on the live meet and then they'll open it up and take it at the same time as you. So we'll take it during class, but I think you will probably, I'll take it on the computer for tomorrow because like I said, I just can't think enough to design anything new right now. So that's my plan for tomorrow, unless I come up with something brilliant before then. Other questions? Okay. So for the cell cycle regulation packet, those of you that wanted to do things on paper, I gave this to you on paper, just mainly because it had all the vocab and stuff for labeling up there. It's up to you if you'd rather do this one on the computer. That's totally fine with me. But if you want to just go through and write the answers on paper so that you have this as part of your notes as well, that's fine. If anybody else wants a paper copy of it, that's fine. People at home, go into Google Classroom, go to that same post where you did your mitosis activity yesterday and work on the cell cycle regulation packet in there. Once you have both the assignments on that post finished, then you can attach them and turn them in for completion points, okay? So folks at home, you are welcome to sign off now and just go and spend some time working on that. If you want to stick around, if you have any questions, you are welcome to do that as well, but you don't have to. They hop off real quick as soon as I say that.
Double shots.
No, it's just a diagram to look at. Yes.
So what do you think?
And of course, you guys can and probably actually should talk to each other about this one. It'll make more sense if you talk through it. A lot of the questions in this just ask you to look at the diagrams to figure things out. So if you're not sure, don't go and Google stuff. See if you can figure it out from the diagrams that they give you.
The first one? Yeah. Because it says that, so let me see, I don't know, or is that G1? Is G1?
No, it's saying that time to move past the checkpoint. No, it is wrong.
Oh, no, it's G2.
Oh, it's G2. Even though it is not in Rome.
Yeah, so like it moves past one, so
That's what I would think, yeah.
You will not be able to go.
Oh, does it attach this window?